Thank you for introduction, and thanks for inviting me to share what we can do in Palmerston North. So those ones who is outside of New Zealand, Palmerston North is 2 hours away, north, that's sort of that way. And it takes 2 hours by car and also by train, so it's not far away. And we do have capability in nucleic acid chemical biology, where I'm really become interested like more than 20 years ago. And our group is rather small, but I think we were quite productive in recent years. And that's us. We are combining chemistry capability. We're combining structural chemistry and biology with biophysics, including NMR experts and cell biology. So myself, I am a DNA and RNA chemist, trained by that, and I have 2 DNA synthesizers I'm really proud of. And we can synthesize DNA and RNA modified chemically. So this is one of these examples when we have a 4 nucleotide sequence cross-linked, and 3/4 of this structure is chemically modified. We can do it in a large scale. The largest scale we produced was probably a couple of weeks ago was 150 milligrams of synthetic oligonucleotide. And we can also do long DNA and RNA sequences in our facility. So we do also have capabilities to study DNA and RNA structures using a suite of instrumentation, including 700 megahertz NMR, the largest field NMR in New Zealand. And using all these biophysical techniques, we can actually pinpoint a lot of things, what happens in the DNA and RNA sequence, how it folds, how it interacts with DNA and RNA and with the proteins. And specifically, we have quite a good structural biology and structural chemistry capability. So we do work with in Australian synchrotron and use small angle X-ray scattering. This is an example when we look at the scattering of the DNA with the protein. But recently we also learned that we can actually use this technique quite well to characterize binding of the small molecules, also to show DNA and RNA sequences. And also crystallography gives us a very good understanding how DNA, sort of in that case, DNA binds to the protein. So I will probably illustrate all our capabilities in the project that we worked in the last 10 years, and the project is devoted to the protein called ApoBec3. So ApoBec3 enzymes, we have 7 of them, humans, and they're actually double-edged swords, because in one case they're actually very helpful for us. They protect us from viruses and pathogens, because they do deaminate cytosine to uracil, and in this way they can scramble DNA of viruses and get rid of them. A lot of viruses do learn how to deal with these enzymes, and that's why they bypass them. In cancer, it was proposed, when these enzymes were discovered approximately 21 years ago, it was also proposed that these enzymes, as a part of our immune system, can actually make a big damage in cancer and contribute to evolution of cancers. And that can lead to development of drug resistance and metastasis. So out of the 7 enzymes, 2 do have a big role in cancers. It is ApoBec3A and B. They do localize in the cell nucleus, and they do have access to the nuclear DNA, and that's where they do the damage of C to U. And they particularly target the dinucleotide sequence TC. And nowadays, we can actually distinguish between the activity of these enzymes, because ApoBec3A really likes pyrimidine in this sort of -2 position, and A3B has some preference for purines. Both of these enzymes contribute to cosmic mutational signatures 2 and 13, which are found in patient samples in patient tumor samples. So these enzymes is now recognized as the largest preventable source of mutations across all cancers. It is even larger than homologous recombination. They are overexpressed in large variety of cancers, and they do also alter cancer driver genes. And the expression of these enzymes correlates with adverse prognostic in breast and lung cancers and reoccurrence after treatment for luminal subtypes of breast cancers, for example. I think this is the most powerful example of activity of these enzymes in detrimental outcomes in cancer patients. It was published last year by Michael Lawrence and Aaron Hatta in Nature. So this is sequencing data for a single patient with non-small cell lung cancer. Initially, the tumor didn't have any ApoB3 signature mutations, but every time the person was given tyrosine kinase inhibitors, it gave rise to expression of these enzymes, and every time the resistant clones had ApoB3 signature, which led to the development of drug resistance. So our proposal in the field is that we would like to develop inhibitors of these enzymes so that we can suppress cancer evolution and allow these drugs to work for much, much longer, ideally getting rid of cancer entirely in the first instance. So one can argue, that in some cases these enzymes might be very useful because they generate a lot of neoantigens. But it's also known, and that may be very useful for immunotherapy. But it's also known that patients who are given immunotherapy, they also do develop drug resistance. And that's, I think, one of the things that we should think about is maybe we should need to keep the genome at the beginning of treatment stable. So there are no mutations occurring, and then we will allow also immunotherapy and targeted therapy work for much, much longer. So how do we develop our inhibitors? We target the active site using inhibitors of cytosine to U conversion. We modify DNA around the cytosine or modified cytosine. An idea is that we can obtain a small molecule inhibitor that is, we can change properties, we can adjust lipophilicity, that we can adjust delivery, so it will look like a drug-like molecule. So in several years, we have actually synthesized several modifications of the nucleobases. We call them zibularins, fluorozibularins, and these 7-member rings. And once we introduce them into the DNA, they become inhibitors of these enzymes. But inhibitors are quite lousy because it's only like inhibition constant in micromolar level. Situation changed when in 2017, it was published the structure of DNA bound to these enzymes. We could see that there is a cytosine in the active site close to zinc. There's a thymine sitting here, and these two nucleobases, A and T, they actually face each other. And the whole DNA is not linear. It actually adopts this U-shaped structure. So we proposed that if we can mimic this U-shape, we can develop much more potent inhibitors. And indeed, this is the case. And we tested that using 2 different approaches. One of them is to use DNA hairpins with short loops, or using a cross-linked DNA where we cross-link distant nucleotides with each other, and we also develop inhibitors. And cross-linked actually behaved much better than the DNA hairpins in this case. So important thing is actually to compare Km value with Ki value, because that gives us an estimation of the apparent affinity of our inhibitors to the enzyme. And as you can see, for the cross-linked DNA, our inhibitors bind 1,000 times better than the native substrate. And importantly, that we actually developed inhibitor for one enzyme, ApoB3A, because activity against ApoB3B was not so good. So we can actually create selective inhibitors, which is also important. So a very elegant experiment was done in the laboratory of our collaborator, Ruben Harris, in the United States, in Minnesota at that time, with Amanda Reffer. And she used an assay in which a defective GFP gene was stably integrated in the chromosomal DNA of HEK293 T cells. And she used a defective Cas9 system with guided RNA opening up the site on double-stranded DNA. And then A3A, freely expressed in cells, comes, changes the C to U, and restores GFP. And in this way, we can see increase in the fluorescent signal in cells. Very pleasing for us, we saw that by adding increased concentration of our inhibitors, in this case hairpins, we saw a decreased deamination efficiency of ApoB3 enzymes. So this is the first demonstration that we can develop molecules that can inhibit these enzymes directly in cells. So why hairpins are so good substrate and inhibitors? It is because, and this is actually, we were able to answer using our recent crystal structure. And in this crystal structure, we could see that our FDZ, that we call it, sits in the active site. There are changes in the chemical structure. But critically, and that's very interesting, that there is this histidine of the protein that forms hydrogen bonding interaction with the nucleobase, thymine. And this kind of artificial base pairing between histidine and thymine becomes the part of the stem, which largely explains why these hairpins with short loops are such good inhibitors of these enzymes. And it also, this histidine hydrogen bonds with thymine, and that also explains the preference of these enzymes for the pyrimidines in this position for the deamination of the sequence, because purines don't have this carbonyl. So this information was critical for us because in the last year, it helped us to design even more powerful inhibitors by introducing chemical changes in the sequence. And we also were able to back it up, this data, using even several crystal structures that which we obtained in the last 6 months. So our next experiment is to test if our inhibitors can delay tumorigenesis in the mouse model in which human ApoB3A is expressed. Mice doesn't have or has only one ApoB3 enzyme, which sits in the cytoplasm. It's not carcinogenic at all. So there is one important, one of the differences in immune system between humans and mouse. So that will be a key experiment, and I really hope that RNA platform can help us with achieving this important goal. So lastly, I would like to thank my team, but I would like to mention that 4 years ago, before the pandemic, we organized a symposium, the first nucleic acid chemistry symposium. This is our international speakers, Rakesh, Tracy Bryan, and Naoki Sugimoto. And this year, with the help of the RNA platform, we organized a second meeting, and a lot of you have been in this meeting. This year in July, in June, and I think we had a very productive 1 day of interesting talks and interesting conversations. So thank you very much for supporting these events, and for people coming there, and for RNA platform for providing support for this. So thank you very much for your attention, and I'm ready to answer any questions.
Thank you so much, Vyacheslav. That was awesome. I don't know if we're all running out of puff by the end of the day, but if there are any questions. Yeah.
I was just curious with trying to target drug resistance in cancers. How do clinicians view that as a tool? Because it's something that we've been thinking about as well, and whether it's worth pursuing. But we've heard it's challenging to go into the clinic with that as a target.
So I think clinicians clearly realize that this is a challenge because they give drugs, the drugs work for some time and then stop working. It is a very challenging space to enter the clinical trials because nobody has done it in the past. I had a good conversation with Sarah about it, about the liquid biopsy, because this is something that we have to really carefully think how, once we have a powerful molecule, how to go into the clinical trials. Because we have to carefully choose patients and monitor the progress so that even within short time frame, within 6 months or within 9 months, would be able to say, is the therapy effective or not? It's a big challenge, but I think we have to face it. We have to go for it. Other questions? No?
If not, we'll just thank Vyacheslav once more time. Thank you so much. So the last